Enzymes. Cute little things. At least I enjoy my subject, don't I? Okay. So let's have a little bit of a knowledge check to see what you guys remember or know. Which of the following is true? Katie.
A.
Who agrees? Do we all agree? Oh, raising your hand to agree or to disagree?
To agree.
Are you all sure? 100%.
No.
Okay, James. Why did I call you James? Jason. Jason. Why did I go?
I'm not 100% sure because there's like 8 other options.
Well, that's a good answer. Hold that thought. Okay, so you all agree that B and C are incorrect. So B enzymes are made up of amino acids only, not necessarily. Sometimes you need something we call a cofactor or a coenzyme. So we have different names. When we say apoenzyme, that means it's the protein portion and it might not be active as it is. It could be active or it may not be active. Once we have a cofactor, it becomes the whole enzyme that is active now to have a substrate and for reaction. But again, not all enzymes require a coenzyme to be active. So that's the enzyme is the active enzyme, and the apoenzyme is the protein portion before a coenzyme, and the holoenzyme is the enzyme that is whole, complete with a cofactor and active. Okay, when the substrate concentration is much greater than Km. First of all, do you remember what Km is? I know we've got over this in food chemistry and even in food analysis and in chem reactions as an undergrad here. But some of you have also got it in what? Biochemistry too? Huh? Yeah? So Michaelis-Menten constant, good. But what is it? What does it indicate? Anybody remembers this? It's okay if you don't remember. We're going to talk about this today. But anybody remembers? You remember by it? No.
It's the constant.
It's a constant, but what is that constant? Anybody remembers?
Substrate concentration.
And the maximum.
At Vmax,
Maximum rate, it
half,
is
half.
Vmax by 2.
Yeah, half the Vmax, and it. If I have in a question for you, which I like this question very much on a test or a final, define Km. So if you just tell me Michaelis-Menten, that is very slight. You will get a very small portion of the grade. You need to say it is the concentration of the substrate at Vmax over 2, and you also need to say that it indicates the affinity of the enzyme to the substrate. Okay, so this is how you need to describe Km, and we'll talk about that all in detail later. Okay, so now that we kind of have an idea of what Km is, I don't know if you remember any of that, or if you know any of that, or if you want to take a wild guess. Go ahead.
Take a wild guess of A.
I mean, it's a really good wild guess, but anybody else, huh? Do we know what first order versus zero order is? When we say first order towards the substrate or first order, what is it? What do we mean?
It is depending on the concentration.
Dependent on the concentration. When we have the substrate concentration much greater than Km, that means we saturated the system with substrate. So now the reaction is no longer dependent on the substrate concentration. It would be dependent on the enzyme concentration. So this one is not, it's zero ordered with respect to the substrate. It's first order with respect to the enzyme. And we will talk about that most soon.
What was the right answer?
Huh?
What was the right answer?
B. Just B. Because this is zero order with respect to the substrate, and it is dependent on the enzyme, so it's first order with respect to the enzyme. Because now the system is saturated with substrate, you have enough substrate to move the reaction forward, so it won't be dependent on the concentration of the substrate, it will be dependent on the concentration of the enzyme. Okay. Can you think of any importance of enzymes in food systems? Why do we care about enzymes in general in food?
Makes meat more tender.
Makes meat more tender. Processing tool. Great. Any other example? What enzyme do we use for that?
Caffeine.
Huh?
Caffeine.
Capain?
Well, it's inherent in the meat, so I don't know what you're gonna add to it.
Oh, okay. So papain and bromelain usually are used. Okay, good. What other example as processing tool or something else?
Clarified juices?
Huh?
Clarified juices?
Processing tools. Yes, clarifying juices by adding pectinase. Okay.
They can contribute to like rancidity if they're in the food.
Food quality. They can contribute to rancidity. They can contribute to enzymatic browning, flavor development through rancidity and other pathways. Okay, great.
Chopping of a specific, chopping the longer molecule specifically.
Okay, chopping would be good as a processing tool. Like think of high fructose corn syrup, for example. Take the starch from corn, chop it down all the way to glucose, and then you need another enzyme, which is to make it a fructose. What do you use? Invertase, which is an isomerase. So it converts glucose to fructose. Perfect. We're talking about all processing tools. We use enzymes in food analysis to determine concentration of certain substrates, like glucose concentration, lactose concentration, maltose. So it's used for analytical purposes. Let's see, what else can you think of? Why do we measure alkaline phosphatase in milk? Remember?
Delineation?
Huh?
Is that the one where it tells if it was properly processed?
Yes, to test pasteurization. Because instead of storing and doing the microanalysis, which takes a couple of days, what you would do is you would test the activity of a thermally stable enzyme that is more stable than pathogens. If it is inactivated, then all your pathogens are inactivated. Let's see. Measuring activity here, I'll show you what I have. Measuring activity, so sometimes you measure alpha amylase activity in flour, and this would determine whether the starch potentially lost some viscosity in application, because if you have high activity of amylase, there's a potential for starch to be broken down, and then you will have less viscosity in a product that might require high viscosity. So you measure alpha amylase. Sometimes you want to know how much enzyme you add for a certain application. So cheese, how much rennet do I need to add in order to obtain a certain amount of curd in a certain amount of time to be most efficient? So I need to determine the activity of rennet so that I know how much to add to obtain a certain output. We talked about quantification of constituents. Sometimes we need to quantitate inhibitors that could be potentially a problem if they're there in the food, like trypsin inhibitor in soybean. When we process soybean, we want to make sure that it is 90% inhibited or above 90% inhibited, because trypsin inhibitor will affect digestion of protein. Also, there's amylase inhibitor. So in order to determine the presence of inhibitors, how do we determine that? Is by running an enzyme assay to see if the enzyme will get inactivated or inhibited in the presence of an extract from a sample. Okay, waste management. So if we have waste streams that are potentially high in protein or high in carbohydrate or high in lipids, they might clog our waste drainage. So sometimes you treat the drains with enzymes, just chopping those large polymers so that you don't have those blockage. So there's so many. We just gave few examples in each category, and we will be giving more examples later. But they are important, basically. Because they are important, enzymes over the years have been isolated and characterized to determine their activity, their uses, their potential problems that might cause in food or potential benefits. So we're going to learn about enzyme kinetics because I know you've seen it many times, but it's very important for us as food scientists when we work with enzymes. We really need to know what parameters impact our enzymatic reaction, whether to promote enzyme reaction, like as a processing tool, or to inhibit enzyme reaction, like we don't want polyphenol oxidase to cause browning in our food. What are ways to inhibit that? So in order to understand how to use them or inhibit them, we need to know something about their enzyme kinetics, which means the activity, the rate of their reaction, how they're inhibited, what are the factors that cause an increase in the rate or decrease in the rate of the reaction. So Vmax, Km, for example, are kinetic parameters that we will be looking at. Okay, so enzyme definition. What are enzymes? The cute little things. What are they? No, all of you know this. Yeah, thank you very much. They're biological catalysts, so they speed up the reaction. How do they speed up the reaction? Lowers the activation energy. There you go, you're done. You can leave now. So, enzymes are biological catalysts capable of specific activation and conversion of substrate. Often your enzyme has high specificity. Some enzymes are more specific than others, but you need to have a certain specificity for the reaction. So there is an enzyme specific to a substrate. So a protease will not hydrolyze lipids. Okay? They will only break down proteins. But within protease, there are different specificity of protease that would target different subunits within the protein. But different enzymes have at least a certain level of specificity to a substrate or a group or a category of substrate that will allow the conversion of that substrate to products and then end up with reusable enzyme that is regenerated to proceed with the similar reaction. So they don't die out unless you inhibit them by heat or pH or inhibitors. Okay, let's look at the story of proteins. Are enzymes proteins? So it's a long story, but before 1920, it was generally thought that they were proteins. And then a split in opinions occurred after 1920 because you got very two smart scientists that worked on enzymes. Okay, I'm going to give you a story now. So Willstetter is a German scientist who isolated peroxidase enzyme and isolated it to a point where it was very pure and it had really high activity. So they tested the activity of their isolate, it's very high peroxidase activity, and they tested for protein content and they found 0 protein. They couldn't detect protein. So then they concluded that protein is not an enzyme. And then later on, Sumner, who was a scientist from Cornell University, isolated another enzyme, urease, and they were able to crystallize that urease and determine it was a protein. So later on, obviously, who won? Scientists from Cornell, because they were able to prove their theory and got the Nobel Prize for it. End of debate. During the debate, obviously, all the European scientists sided with Willstetter against the American scientists. But for once, American scientist won. Not for once, I'm sure there were many times. But in this case, that was the end of the story. Now, are all enzymes proteins? What about ribozymes? They are RNA molecules. And here, this group of researchers, I put a paper up just for reading if you're interested. They looked at the activity of ribozymes in biological cells, and they have a catalytic activity to convert or to build protein or to catalyze certain reactions in the biological cell. But in foods, we don't care about ribozymes. So food-related enzymes are all proteins. Okay, just wanted to throw that out there. All right. So starting with the 1930s, or actually before in the 1920s, scientists isolated protein enzymes, but it continues through the 30s and onwards to characterize these enzymes and to determine their activity and usage. So in order to isolate them, purify them, many different techniques were used: ion exchange, size exclusion and affinity, electrophoresis, and also techniques based on solubility, differential solubility. Similar to the example I gave you when we talked about protein separation, I was isolating an enzyme and an enzyme inhibitor back at Purdue. So based on differential solubility. So over 3,000 enzymes have been purified so far and structure elucidated. So the first enzyme with the primary sequence elucidated was ribonuclease. And then further other, obviously, enzymes, they were sequenced. Determining the sequences was either by gene sequencing or MALDI, and then determining the secondary and tertiary structure by NMR. And they were found that some enzymes are homogeneous or heterogeneous. That means either made up of amino acids only or other components. And also, enzymes can be synthesized using PCR once we know the gene that actually is coding the amino acid sequence of those proteins. Okay, the other overview I would like to just share with you, because so that when you hear the name of enzyme, you know what it does and what it functions. So there is the Enzyme Commission that sometimes you get an EC number, that's for Enzyme Commission, for an identification of the enzyme. So each enzyme that is registered will have an EC number. Okay, that's the number. And then you have a trivial name, a commodity kind of commonly known name. So trypsin, papain, pepsin, bromelain, these are all catalase. These are all trivial names. But the scientific systematic name, you will see the name of substrate, and then it ends with an ase, but there are different categories of the enzyme. So oxidoreductase, for example, hydrolase, transferase, lyase, isomerase, and ligase. So what are these different categories? So oxidoreductase, example, hydrogen peroxide oxidoreductase, which is catalase. So it catalyzes an oxidation reduction reaction, conversion of hydrogen peroxide into oxygen and water, for example. Okay, hydrolase, it involves water, hydro. So there is water plus the enzyme plus the substrate gives you the product. So think about triglycerides. So triglyceride plus H2O plus lipase, triacylglycerol lipase, would break into 3 fatty acids and glycerol. So you need the water components, OH and H. No, I can't talk. Excuse me. Okay. So you need the water components in order to form the product. So the water components take part in the formation of the product. Transferase will be an enzyme that will transfer a group from one substrate to the other, like D-glucose 6-phosphotransferase. So this takes a phosphate group from ATP, for example, and giving it to a glucose. Lyase, it would be a breakage of a double bond, or without water being involved, so breakage of a double bond or formation of a double bond. Isomerase, conversion of one isomer to the other, like the invertase. So, is it invertase? No, invertase is sucrose to glucose and fructose, isn't it? Sucrase is invertase, isn't it? I thought so. Okay, I need to look that one up. Okay. But anyway, isomerase, changing one isomer to the other, like glucose to fructose. And ligase is formation of a covalent bond using ATP as energy source. So, so this is the example of when you have the substrate and the category of the enzyme and the trivial name. Same thing, glucose 6-phosphotransferase, that's your substrate, and transferase is in the name. And triacylglycerol acyl hydrolase is the enzyme that hydrolyzes triglycerides, and lipase is its trivial name. Okay. All right. So, enzyme specificity. So again, highlighting what I said earlier about in order for the enzyme to work, there should be some specific interaction between the enzyme and the substrate. So the enzyme must bind with the substrate stereospecifically and correctly into the active site. So there is the active site here, this is your enzyme, this is your substrate. So they stereospecifically, based on the structure of the substrate and the structure of the enzyme, there is a location where they can bind very specifically, resulting in lowering the activation energy and converting a substrate into a product. So rate is affected by removal or small change in the configuration of the group. So the rate of the reaction, that is, it's impacted by the change in the structure, that is, of the enzyme or the substrate. If there has been any change, then it will impact how they bind to each other. Do you think the binding is covalent or non-covalent? The type of interaction between the substrate and the enzyme. Huh? Non-covalent, because it's a reversible reaction. So enzyme, actually, when the reaction happens, the enzyme binds to the substrate, and there's two pathways. So the enzyme to substrate can convert to enzyme and product, and can also go backward to enzyme plus substrate. So it is a reversible. So if it's a reversible interaction, it won't be covalent. It would be interaction via hydrogen bonding, hydrophobic, electrostatic, dipole, van der Waals, etc. Originally, it was thought to be, the theory was lock and key theory. So that means it's not only stereospecific, but the active site is rigid. It doesn't change. Once it is, the substrate is there, there's the lock theory, the key and lock theory. But this theory was debunked later on, and it's the more accepted theory is the induced fit. So the active site will change slightly as the substrate binds to it. So it is not a rigid site, and also it doesn't only change as the substrate binds to it due to the chemical environment of the substrate and the enzyme together, it will also be impacted by the environment. So that's why, for example, activity of the enzyme is different under different conditions. So if under different pH, there will be different ionization occurring with the enzyme. So the structure and the configuration of the enzyme will be slightly different, especially with the active site. So the pH, the temperature, the ionic strength, water activity, they all impact the structure of the protein, which is the enzyme. Therefore, hence impacting how this will specifically bind to your enzyme. Not stereotypically, stereospecifically. So again, that's why we have optimum pH, optimum temperature, optimum ionic strength, and the water is very important, the presence of water and the water activity. Okay. So the minimum of 2 events happen. Actually, there are 3, but the minimum of 2 events is when enzyme binds to the substrate to give you the ES. And there's the rate, K1 is the rate of the first reaction. And then you have the second event where the ES converts to products and the enzyme is regenerated. K2 is the rate of that second event. The other event that could happen, and it's overlooked usually, is the ES actually dissociates back to E plus S. However, this is impacted by the concentration of the substrate. So if you have an abundance of substrate, your reaction moves mostly forward. When you don't have an abundance of the substrate, then the K minus 1 starts going, start becoming larger. And the rate of the forward reaction would be reduced, basically. So this is just an illustration of converting sucrose into the products glucose and fructose. Sucrase is invertase. I'm 100% positive. It's just coming back to me. All right, so this is just another illustration of the activation energy. So without an enzyme, you require a really high amount of energy to convert a substrate into a product. However, given the catalytic function of an enzyme, once it's there, it's going to reduce the amount of energy needed to drive the reaction forward. So it's going to speed up the reaction and convert reactants to products. So enzymes accelerate reaction by a factor of 10 to the 3 to the 10 to the 11th times that of non-enzymatic reactions. So that's how fast, how enzymes can speed up the reaction. So talking about speeding up the reactions, let's look a little bit about the velocity of the enzyme reaction. So the velocity of the enzyme reaction, or the rate of the enzyme reaction, or how fast the enzyme reaction goes. So it is directly proportional, obviously, to enzyme concentration. So the more enzymes you have in your system, the higher the rate of the conversion of substrate to products. It is also dependent on the concentration of the substrate, which we'll talk about a lot, presence of inhibitors, and presence of cofactors. Like I said, sometimes the enzyme, in order to be active or more active, we need cofactors or coenzymes. All of these factors, we'll talk about them individually later on. So it's affected by, obviously, environmental conditions. We talked about optimum pH and optimum temperature, and also ionic strength that will impact the charges on the protein active site. So for instance, for pH, velocity can be decreased by 0.5 times with every 1 pH unit away from the optimum pH. Similarly, temperature, also the rate of the reaction or the velocity decreases 1.4 to 2 times with every 10 degrees difference in temperature, going up or going down. Both pH going up or going down away from the optimum. So with all of these factors impacting enzymatic reaction, we really need to understand enzyme kinetics to control rates of enzyme catalyzed reaction. Now, why is it important? Is that me? I don't know why. Okay. All right, so why is it important? We talked a little bit about applications earlier, and I probably threw in why it's important. Understanding kinetics. Why is it important in food systems? Yeah, okay, you're tired. You had enough of questions.
Rate of oxidation?
Huh?
What?
What? Say that again?
Is oxidation involved?
Well, not necessarily. I mean, there are enzymatic oxidation by lipoxygenase. So if I want to understand lipoxygenase activity to inhibit reacidity due to oxidation reacidity, then yes, I need to understand the enzyme kinetic of lipoxygenase. So we're close there. Anything we talked about, using rennet in cheese making, preventing or inhibiting polyphenol oxidase, measuring or determining the presence of inhibitors. All of this requires that we understand the enzyme, understand its affinity to the substrate, understand its velocity under different conditions. So therefore we can design an enzymatic activity assay, or know how much to utilize of an inhibitor, for example, to achieve reduction in browning over time. So we really need to understand enzyme kinetics for many applications in food systems, and that's why we're talking about it over and over in multiple courses in food science. Okay, so I'm sure you love this graph and you've seen it many times before. So when we design an enzymatic reaction, we are monitoring something. What we usually monitor is production of your products. So we look at the concentration of the product over time. So the first step in any kinetic determination for any enzyme is plotting such graph. Concentration of a product over time. So that's how we start. Okay, and then from there, as we monitor, we have a pre-steady state, and it's a very, very short time. Sometimes we don't even, we're not even able to monitor it because it is few seconds where substrate and enzymes complex. So to go from E plus S to ES, it takes few milliseconds to happen. And then after that, we have rate of disappearance going this way and even that way equals the rate of appearance. And that we call the steady state. And you can see here, we have a linear kind of curve. Here is your steady state, here where you want to use this space to determine kinetics of the enzyme. And the slope of this linear line is your rate, or V0, or initial velocity, which is the rate of your reaction. So, as you monitor products and over extended period of time, you see that it's no longer linear because something's happening here until it plateaus. What do you think is happening so that it's no longer linear? The substrate. What happened to the substrate? We're running out of substrate. So now, some of this is really going backwards now. So it's no longer that much forward. You're not generating as much products until most of the substrate is really depleted. We don't get anything anymore. Okay? So again, it's very important to get initial velocity in the linear range in order to complete the kinetics study. Okay. That's not that. This is your second favorite figure here, which, yeah, I'll let you go. I was hoping that I can cover 3 more slides, but it's okay. We'll do that. After the break, enjoy your break.